Hello students, today we are going to see the second part of HPLC. In the last part, we have covered introduction and basics, scope of HPLC, instrumentation. In that we have covered the various parts such as mobile phase, palms and columns in detail. Now in this part we are going to see about the instrumentation, and in that we are going to see the remaining part, that is the detectors. Now detectors are called as brain of chromatographic system. It helps to record the chromatogram based on certain characteristics of the analyte and help us in identifying that compound by qualitatively and quantitatively. Detector senses the presence of individual components as they elute from the column. The detectors are defined as the thing or the or in part which converts a change in column efficiency into an electrical signal that is recorded by data system. There are two types of detectors. First is the selective detector. It is also called as solute property detector, and another is universal detector. It is also called as bulk property detector. So in the first, selective or solute property detector, it responds to properties of solutes. For examples such as UV visible detector, fluorescence detector or diffusion current detector. These are of two types, destructive detector as well as non-destructive detector. In destructive detector, our substance or analyte is get destroyed and we are unable to recover it. In non-destructive detector, we can recover our sample, such as example is the UV visible spectroscopy or RI, refractive index detector. In this we can recover our sample. Now in the universal or bulk property detector, we are analyzing the mobile phase depending on the bulk property of the compound. For example, refractive index detector, it measures the density or we are measuring the dielectric constant also. There are characteristics of the ideal detectors. We are going to see some ideal characteristics such as versatility. Our detector should be versatile, it should be highly sensitive, it should have capacity to continuously monitor the column effluent, it should have low noise level, wide linearity of response, insensitive to flow rate, it should have good stability and reproducibility, it should use linear response to analysis that have several ranges magnitudes. Then it should give shorter response time, it should be highly reliable and easy to use. It should have similarity in response towards all analysts. It should be non-destructive, cheap, it should be easy to use and able to measure small peak volume. Now we are going to see various types of detectors in HPLC. So first is the UV visible detector. It has of various types such as fixed wavelength detector, variable wavelength detector, diode array detector. Other detectors are such as fluorescence detector, electrochemical detector, refractive index detector, conductivity detector, mass spectrometry, evaporating light scattering detector, FTIR detector. So these are the detectors enlisted here for which is commonly used for the HPLC. Now we are going to see the first detector, that is the UV visible detector. So in the UV visible and PDA detector, these are categorized as absorbance detector. They provide good sensitivity for the light absorbing compounds at the picogram level also or nanogram level. They are very easy to operate and provide good stability. UV detectors are very commonly used detector in the HPLC analysis. The compound which have very strong UV visible chromophore, that is having the double bond or aromatic molecules, will very nicely use the absorbance in the UV. The advantages of this UV visible detector is it is very simple, reliable, inexpensive, compatible with the gradient illusion and non-destructive. disadvantages are it it is non-sensitive, such as it is not as sensitive as fluorescence detector. Means fluorescence detector is more sensitive than the UV visible detector, and it is not universal also. Means only the molecule which have the chromophore, we can analyze that molecule only by using the UV visible detector. Then what type of compounds we can analyze? Example, vitamin, carotenoid, phytonutrient, etc. Those compounds, they are unsaturated. We are able to detect by using UV visible detectors. Now, UV visible detectors are equipped with various components such as filter, monochromator, or it has UV visible detector. We can analyze UV compound by using UV visible detector at fixed wavelength, that is generally available at every institute, or dual wavelength, or diode array detector. Now this is the general detector, UV visible detector. This is the detector it looks like. So this is called as the flow cell detector. During the analysis, sample goes through a clear colorless glass cell. This is called as flow cell. So at the top we are seeing the sample or mobile phase is coming from the column, and it is going to the west, and one horizontal line is there. From there the UV light or UV source is passing, and it is going straight towards the detector. So when UV light is irradiated on the flow cell, sample absorbs a part of UV light. Thus, the intensity of UV light observed for the sample or mobile phase and the effluent containing sample will differ. By measuring this difference, the amount of sample can be determined. A standard UV visible detector allows user to choose wavelength between 200 to 800 nanometer, and the most commonly used wavelength is 254 nanometer. This advantage of this detector it should be highly sensitive. It is highly sensitive. Then small volume is required. Then linearity, it should be linear over the wide concentration range, and it can be used with gradient illusion system also. Disadvantages: it does not work with compounds that do not absorb light at this wavelength region. Now here we are able to see two different diagrams. One is the variable wavelength detector, and another diagram is of the photodiode array detector. So what is the difference between the normal UV detector and photodiode array detector? Generally the common UV detector, it is giving 2D spectra to us, and in the photodiode detector we are getting the 3D spectra. In the variable wavelength or single wavelength detector, we are passing the monochromatic light towards the sample, and detector is detecting the transmitted light or transmitted light obtained from the sample. But in the photodiode array detector, we are passing the wavelength of light, all wavelengths of the light, not only the monochromatic light, but all wavelengths of light we are passing through the flow cell or the sample, and detector is detecting all the wavelength one by one, and like this we are getting the 3D spectra by this type of detectors. Now this is the graph or diagram which we are seeing over here is the 3D spectra. So this diode array detector work very parallelly or simultaneously and monitor all the wavelengths and gives the 3D spectra of the sample. This is the spectra obtained from the normal single or dual wavelength detector. This is the 2D spectra. This we are complete the UV visible and PD detector. Now we are moving towards the fluorescence detector. Now the fluorescence detector. The advantage of fluorescence detector is it is highly sensitive for selective group of compounds at femtogram level. By using a specific wavelength, analyte atoms are excited and then emit light signal, that is fluorescence. The intensity of this emitted light is monitored to quantify the analyte concentration. So the compound which have fluorophores, such as in nature carbamate, pesticide, aflatoxins, vitamins, amino acids are there. So all these have fluorescence. So we are able to detect these compounds by using this fluorescence detector. And if our compounds are not having fluorescence, so we can do post column derivatization, or we can use the derivatives for our sample, such as densyl chloride, and we can analyze that compounds by using fluorescence detector. Advantages: it is highly sensitive, then it have low background noise, it is highly selective also, and it can solve two co-elution problem. Post column derivatization is also possible with this detector. Disadvantages: it is difficult to use, then more instrumental variables are accounted for during optimization, or changes in fluorescence if pH or viscosity change is possible. So these are the disadvantages. Now what type of compounds we can analyze, such as vitamins, drugs, aflatoxins, etc. we can analyze by using the compounds, or some natural products or clinical sample, petroleum samples also we can analyze. So these are the advantages we are seeing. This is the diagram of the fluorescence detector. Here excitation wavelength is there, then detector is there, in between sample is there. We are seeing this in the last scene also. So the arrangement is at 90 degree. For this means scattering of light we are observing over here, that's why the detector is situated at 90 degree. Now we are going to see the next, that is the electrochemical detector. Electrochemical detector, here the detection is based on amperometry, polarography, colorimetry and conductometry. They offer high sensitivity, simplicity, convenience and widespread applicability. It is easily suitable for use with semi-micro or capillary tube system. So electrochemical detectors, in this we can account polarography, amperometry, colorimetry and conductometry. These are this is running on the base of this principles. Electrochemical detector advantages are it is highly sensitive and easy to use. Disadvantages are mobile phase must be conductive, mobile phase must be purified from oxygen, metal contamination and halide respectively. Now example of electrochemical detector is the conductivity detector. So conductivity detector, in the solution containing ionic compounds will conduct electricity. Conductivity detector measures electrical resistance and measured value is directly proportional to the concentration of ions present in solution. Thus it is generally used in ion chromatography. The electrical conductivity detector measures the conductivity of mobile phase. There is usually background conductivity which must be backed off by suitable electronic adjustment. If the mobile phase containing buffer, the detector gives a base signal that completely overwhelms that from any solute, usually making detection impossible. Thus, the electrical conductivity detector is a bulk property detector and sense all ions, whether they are from a solute or from the mobile phase. in order to prevent polarization of sensing electrodes. AC voltages must be used, and so it is the impedance, not the resistance of the electrode system, that is actually measured. From a physical chemistry standpoint, the conductivity of a solution is more important than its resistance. However, it is the resistance impedance of electrode system that determine the current across it. This is the diagram of electrical conductivity detector. Here outlet and inlet is there, and between that electrodes are situated. The electrical conductivity detector has a sensitivity of about 5 into 10 raised to power minus 9 gram per ml and the linear dynamic range of about 200. It is used in probably over 95% of all analytes involving ion exchange procedure to separate ionic and inorganic and organic ions. Now next we are seeing about the ELSD, that is Evaporating Light Scattering Detector. This ELSD provides a good sensitivity for non-volatile analytes at nanogram level. The column effluent is nebulized and then evaporate to form the fine particles. Now it is targeted for the lipids, sugars and high molecular weight type of compounds. So this detection is based on scattering of beam of light by particles of compound remaining after evaporation of the mobile phase. It is a universal detector and does not require a compound to have a chromophore for detection, such as surfactant, lipid and sugar. Unlike the refractive index detector, which was formerly used for this analysis, it can be with gradient illusion and is robust. Means we can use it for the gradient illusion, gradient mobile phase if we have, then also we can use it for enough function under a wide range of operation. Means it is wide variety of operations also we can perform. However, it cannot be used with non-volatile materials such as buffer in mobile phase or to detect very volatile analytes. Typical application include analysis of chloride and sodium ions in the pharmaceuticals, lipid used as component in formulation, sugar and sugar polymers also we can analyze. This is the diagram of ELSD detector. At the top we are seeing the column effluent, at the middle we are seeing the flame, then nebulizer is there, from one side light source is passing and at another side photodetector is there. So how it works? Generally the column effluent is nebulized. Whatever the column effluent we are passing, it is get nebulized and convert into fine mist by a flow of nitrogen or air. Then due to the nebulization, fine droplets are formed and it is carried through a controlled temperature drift tube where evaporation of mobile phase occurs, leading to formation of fine particles of analyte. The cloud of analytes particle is passed through a laser beam. The scattered radiation is detected at right angle to the flow by a silicon photodiode. Advantage of this ELSD is it responds same for all non-volatile solutes, a lot more sensitive than the refractive index detector. The next is the refractive index detector. Refractive index detector, this refractive index detector measures change in refractive index. This is the diagram of refractive index detector. Here the refractive index detectors measure the change in refractive index. It is more sensitive, it has a lower sensitivity compared to the UV detector. That is the reason it is not as commonly used as the UV detector. The compound that do not have a very strong UV visible chromophore or fluorophore or electrochemical activity or ionic conductivity is measured by using refractive index detector. It is advantages, it is universal in nature. Disadvantage it is lack of sensitivity. It has lack of sensitivity. It is impractical for gradient illusion, and instability of baseline is obtained by this. The sample which we can analyze is organic acids, we can analyze sugar, fungal metabolite, oligosaccharides also we can analyze. Now this is the diagram of the refractive index detector. Here a glass cell is divided into the two cells such as reference cell and sample cell. The effluent from liquid chromatography column flow through a sample cell, while other called reference cell is filled with only mobile phase. When the effluent going through sample cell does not contain any analyte, the solvent inside both cells are the same. While if effluent contain any components other than the mobile phase, bending of the incident beam occurs due to the refractive index detector difference between the two solvents. By measuring this change, presence of components can be observed. The refractive index detector is one of the least sensitive liquid chromatography detector. It is very sensitive to change in ambient temperature, pressure change, flow rate change, and cannot be used for gradient illusion. Despite there are many disadvantages, this is extremely useful detector for those compounds that are non-ionic, do not absorb UV visible light or do not fluoresce. For that it is very useful. Then next is the mass spectrophotometer detector. The analytes are detected based on their molecular weight. The obtained information is especially useful for compound structure identification. However, its use is not limited for structure identification and can be used to quantify a very low detection limit of element and molecular compound. It is the destructive type of detector. Here we cannot recover our sample, but by using this detector we can analyze the molecular weight of the compound. Not it is helpful by using the molecular weight we can identify the fragments also, and probably we can detect the structure also. So it is when liquid chromatography is attached with the mass spectrophotometer detector, it is most termed as the LCMS. So this LCMS is the analytical chemistry technique that combines physical separation capability of HPLC with the mass analysis. While liquid chromatography separates the mixture with multiple components, mass spectrometry provides structure identity of the individual component with high molecular specificity and detection sensitivity. This tandem technique can be used to analyze biochemical, organic or inorganic compounds commonly found in complex samples of environmental and biological origin. Therefore, LCMS may be applied in a wide range of sectors such as biotechnology, environmental monitoring, food processing and pharmaceuticals, agrochemical or cosmetic industry. Next is the FTIR detector. Here in FTIR detector we can separate our compound as well as analyze the compounds. Their functional group we can analyze by using this FTIR detector. So here two types of FTIR absorbance detectors are available. The wavelength scanning is provided by semicircular filter wedges. The wavelength is ranging from 4000 to 690 centimeter. The other Fourier transform type is more sophisticated and manufacturer provides the accessories for use of HPLC detectors. The cells of Windows are made up of NaCl or CaF2. It is not very sensitive and have serious drawback that most mobile phase solvent absorbs strongly in the IR region. Here we have completed all the system detectors, then data system. So we can obtain our data for better accuracy and precision. Data station such as computer is required to record the complex data. Then software is there which with the help of that we can record the data. Then various types of recorders are there such as strip chart recorder to record the retention time or peak areas or peak height. Then integrator is there, and we can analyze our data based on computer control data collection. So here these are some references. Here we have completed all the detectors today, and in next part we are going to see in detail. This is the second part. In the third part we are going to see in detail about the applications. Thank you.